Hello everyone, thank you so much for checking out my video. My name is Gaurav, I'm a fourth year biochemistry major here at RIT, and today I'll be taking you through my past research this summer. I'd like to start off by first giving a special thank you to Dr. Michael Coleman for his generous supervision and mentorship throughout my research this summer. I would also like to thank the Howard Hughes Medical Institution for their generous grants and funding for my research. This summer, my research was about an organic synthesis method of creating NHCs, or N-heterocyclic carbenes, which act as a ligand in organometallic transformations. Although most are known, chiral NHC transformations are a relatively new field. Throughout our research, Dr. Coleman and I put forth a synthesis of a novel NHC catalyst, which will in turn enable sustainable and more efficient economic access toward many complex pharmaceutical ingredients. Keeping that in mind, the direction of pharmaceutical research and drugs in today's society has been leaning more and more towards chiral entities. Chiral, meaning chirality, is a property of asymmetry, which is important in many different branches of science. The word is actually derived from the Greek word "cher," which means hand, and a molecule is said to be chiral when it has a non-superimposable mirror image. So if there's a mirror in between these two hands, as you can see, these two images are called enantiomers, or mirror images of one another. Now the thing about this is they have the same physical properties, but if you try to take the right hand symbol here and put it back on the left hand, it will not be the same superimposable image. Therefore, we say that this carbon is a chiral carbon. Chiral molecules are everywhere around the world. It is an integral property which underlies many of modern organic synthesis. So if you look to the right-hand side here, these are all examples of just some of the vast majority of drugs that contain chiral centers within the world. And you can see the chiral centers by looking for the wedges or dashed lines. So you see a wedge or a dash, and you'll see a chiral center right next to it, which is a carbon which has 4 different groups attached to it. 1, 2, 3, and the fourth one would be the hydrogen off the side. So my question learning this was, if these two different enantiomers, or the two different mirror images of a compound, have the same exact physical properties, why do we care? It turns out that the two mirror images that are formed from a chiral center atom can have major implications towards the outcomes of that substance, especially in a chiral environment like the human body. One example of such is the drug thalidomide. This was prescribed to pregnant mothers in the mid-20th century as a means to cope with their morning sickness. And you can see here, this is the thalidomide structure. The right-hand side enantiomer of the drug, so one of the mirror images, was a bioactive sedative, while the left-hand side of the drug had exposed the mothers and infants to birth defects and had an infant mortality rate of 40%. As you can see, with one enantiomer of the drug becoming a cure, while the other enantiomer of the drug becoming an even bigger problem, this created an uproar in the scientific community. Due to this, the FDA required all drugs with chiral centers to be evaluated separately between their right hand and their left hand sides in clinical trials. During this time, organic chemistry had a sort of renaissance in which they had to address the synthesis of new chiral molecules with asymmetry. In order to do this, more techniques had to be created to synthesize these chiral molecules in a selective fashion for only one hand or one enantiomer of them. Because up until that point in the early 2000s, all traditional organic synthesis techniques made both hands of the product drug. One of such techniques is in the form of creating chiral catalysts. Now, we move 20 years forward into the future, and chiral catalysts have become a staple in the pharmaceutical industry. These catalysts are being used to both test substances and the effect of all their hands based upon the amount of chiral centers present within the substance, and also prescribe a certain specific handed substance to the general public, which is a drug. The normal CNHCs, chiral catalysts that are used, that have been used, you can see at the bottom left-hand corner of your screen, which is this structure. There was introduced a new class of chiral ligands, chiral catalysts, which is the one that we used, which is considers 2 ligands here and 1 metal underneath. We are doing a different class, which has a different shape, as you can see, in order to put less toxic metals into them, which in turn makes them more efficient and more atom economical. This means that whatever products we put into or reactants we put into the synthesis, we do not have many byproducts of the synthesis. On this slide, you can see our novel chiral ligand design with what I said before is this metal that is attached to, so these are the ligands on the outsides, and this metal that is attached could be any of the group 11 metals, which are non-toxic, which will in turn make the reaction more efficient. And as we're using non-toxic and less precious metals than we used before, such as rhodium and palladium, which are very, very toxic and very rare to find. As you can see here on this slide, this is the full chiral catalyst is what you're looking at. So my business this summer was to find the synthesis of one of these ligands. So this is a synthetic design that we proposed, and it's a basic organic synthesis design using traditional building blocks that we have as we've made these building blocks earlier in the lab. So how do we set up this reaction? Well, the reaction setup can be seen on the left-hand side with this picture. This is a syringe pump with syringe full of reactant leading to a round bottom flask that is purged with argon. So basically, in order to create this non-toxic, non-precious metal containing chiral catalyst, we actually had to use a toxic and precious metal in the workup. We used a solid form of rhodium, which is very toxic and precious. So Dr. Coleman and I decided that the best way to go about it was to do 5 separate organic synthesis reactions and add them all together at the end. So basically what happened was the rhodium along with hexane and diphenylethylamine were added into this round bottom flask as it was stirring. And the rhodium is actually a solid, but this way it would dissolve in the solution. And then the diazoacetate, which is the other building block that you saw on the last page, would be added here, also with hexane, into this syringe, which would then be pumped by the syringe pump. It's basically a machine that pumps the syringe into our reactants, or into our reaction rather, at a steady rate of, we use the rate of 2.67 milliliters per hour. So this reaction total was done over the course of 3 to 4 hours and was left to stir overnight. After that, the next day, it was concentrated in vacuo by rotary evaporator and was then also vacuum evaporated. That was in order to get rid of all the solvent, all the hexane that we needed to use in order to dissolve everything. So after the reaction was completed, we had to get rid of all of that solvent. So as I said before, this reaction was then repeated for another 4 times, so for a total of 5 reactions, and the products of all of those reactions were then all placed together into one round bottom flask to create a master mix, crude master mix of all our products of those reactions. Then those products were analyzed, and the first type of analysis that was performed was NMR, nuclear magnetic resonance spectroscopy. So this is basically a tool. This is the NMR at RIT. This is the tool which uses a magnetic field to a nucleus, and we use the 1H, or the hydrogen isotope 1, and also a radio frequency pulse, which characterizes the frequency that resonates from your product within the small tube that you place into this NMR machine. And then from that, it can tell you, since we're doing the 1 hydrogen isotope, it tells you all of the hydrogens that it finds based on the resonance frequency pulses. It tells you all the hydrogens it finds within your product, and it also will tell you the environmental surroundings of your hydrogen, so that in that way you will know which hydrogen is where and which hydrogen is what when you see it on the NMR. So this is what our crude NMR looked like for the master mix of our 5 different reactions. And so the peak right here would be our starting material, all the hexane, everything else. These peaks would be the solvent that we needed to put the solution in order to even measure the NMR. And these 3 peaks are our product peaks. There's one singular peak and there's 2 doublet peaks, which is why you see 2 different numbers for each of these. These are the hexane peaks, which is the solvent that did not fully come off after vacuum. So this is our crude NMR of our reaction mix master 5 times reaction. Another type of analysis that we used in order to help purify our compounds was chromatography, both flash chromatography and thin layer chromatography. With thin layer chromatography, we ran plates, which look like these, and each spot that shows up under UV light would be what we had in our sample. So this was the baseline control product that we found from the racemic mixture. And these are all the, from 1 to 24 of the fragments that were created by the biotage in flash chromatography. These are all the impurities and products that we found. TLC plates separate our impurities and products by way of polarity. The most nonpolar products will run up higher upon the TLC plate, while the most polar products will rise slowly up or maybe not move at all. Through the biotage, different fragments were created and collected, 1 through 55, which again is flash chromatography, which also separates the fragments and aliquots by way of polarity, most nonpolar versus most polar. And through this, we took TLC plates of each sample, as you can see here, and we found that we needed to use a certain type of them, which had the same amount of product as our final. So through this, we were able to purify our product, and this is what we had through NMR. Again, this is our solvent that we needed to use by the NMR in order to do it. As you can see, the starting material, the peak at 5 is gone, and we only have our products left along with some water. Now that we had found out that we had a pure product, we needed to run an chiral HPLC. HPLC, or high performance liquid chromatography, is a method which is used again to separate compounds, mostly in analytical chemistry, and it is also used to quantify the individual components of the mixture. The separation of our mixture happened by using a chiral column medium in which components move at different rates. Again, chirality is different hands. So one hand would then move faster through the column than the other hand would. So we would then know if we had 2 hands or if we had 1 hand, but we would not know which actual hand that we had in the end. After learning all of this and setting everything up, we ran our chiral HPLC with our purified product, and we got these results. Now, the figure on the left shows the results for the racemic compound that we started with, which contains both hands of our goal substance. As you can see here, the enantiomeric excess of our compound was calculated by determining the area under the graph of each of the huge peaks. As you can see here on the left side, there is a huge peak at around 4, which goes up to about 600. And there is also a peak at 17.5, which goes to about 100. As you can see here in the chiral HPLC results for our compound, rather than the racemic, we still have that peak at around 4 or 3.6, 3.7. This solution is much more diluted, so it was only at 40. But we, again, as you can see, we do not have the peak at 17.5. So this is showing that we have 2 hands in our racemic mixture, 2 hands, while we only have 1 hand here. So this shows us that we did indeed create only one enantiomer of our compound, and it is pure. The physical results we then obtained from this information was our enantiomeric excess, which shows us how much of one enantiomer we have versus the other one. And it also showed us the amount of yield we got based on our actual starting reactions and products. The yield was afforded 88%, and we had a greater than 98% enantiomeric excess, as you can see, by no peak at all at around 17.5. In the future, we're going to use our derivation of compound 1 and create another organic synthesis reaction in order to add it onto compound 2, which will in turn create the whole chiral catalyst. I would like to thank again the Howard Hughes Medical Institute and their Inclusive Excellence Grant for their generous summer support and funding through my research this summer. And again, Dr. Coleman for his mentorship and guidance throughout this whole process. It was a very eye-opening and fun experience for me, and especially as I'm looking forward to pursuing my doctorate in chemical engineering. Thank you everyone for watching. I hope you enjoyed.